Well, my name is Gerardo Medina. I'm a postdoc/research associate at Dr. MacKenzie's lab. I just started 2 years ago with this amazing project that Alex had the audacity to leave it to me to just try to make some sense on what we're having here. So before I start, I can see a lot of stressed people because Dr. Rami just gave us a test. So let's do a little bit of an exercise. Let's breathe out, breathe in, breathe out, breathe in, breathe out. Well done. That helped me with my nerves, so that's fantastic.
Sorry, I didn't mean to
So first, I would like to thank the SHINE Syndrome Foundation for the opportunity to give this talk and also to be in front of all of you. So today, I'm going to be talking about our most recent findings looking for a therapeutic approach for the SHINE syndrome. And our work bridges molecular neuroscience, nutrition, and translational research. And we consider this to be an important work, not just because of the science that we're doing, but because it's beneficial for those families and individuals with SHINE. So I'm not going to be touching here much because it's just, again, once more and more and more about what the SHINE syndrome is. I just wanted to take away that we're going to be focusing on the loss of function of the DLG4 copy and with a loss of PSD95 function. So, and this one, same, we'll be talking a lot about PSD95. So the only thing I'm going to mention is just follow with what Dr. Thomas said. Since PSD95 is the mother of the postsynaptic region, I'm just going to go a little bit further and give it a job title. So I think that PSD95 is not just part of the structure, but also the architect of the synaptic communication. Because when there's a mutation on the DLG4 gene, there's a disruption of the PSD95, which also controls the communication. Therefore, there's going to be an impairment on this communication, or the connection between the neurons. So for that, rather than focusing on a replacement of the gene, we decided to ask a different approach, a different question. So can we find compounds that well-tolerated and clinically approved compounds that can increase PSD95 levels, and if so, also include an increase on their function? So for that, we decided an experiment looking at differentiated shy-wide neurons. These are a neuroblastoma model that produces endogenous PSD95. We use immunofluorescence to determine the quantification of PSD95 as our primary synaptic marker, but also we look at MAP2 as everyone has been doing as well, because it is an important dendritic structural protein. So it helps us to look at the neuronal health and morphology for the neurons. For every drug that we tested, we used 3 clinically relevant concentrations. So what are these drugs? So we went into the literature. We were looking at those drugs that can upregulate PSD95 and also that can upregulate or regulate the dendritic spines. So we, in collaboration with the Medicine Precision Institute in the University of Alabama in Birmingham, we come up with a curated list looking at their effect, their potential to be both scientific and translational drugs to use. So what we found is that out of all the compounds that they can moderately upregulate PSD95, we saw that DHA, an omega-3 fatty acid, was capable of increasing PSD95 levels after 24 hours significantly compared to control. It went up to 1.7-fold. And similar result was observed after 72-hour exposure when we were looking at longer exposure potential as well as safety for users. We also found that ketamine, allopurinol, valsartan, and fluoxetine were able to increase the PSD95 levels, but more moderately compared to DHA. So now looking at the neuronal health using MAP2, we also observed that a lot of the compounds, instead of upregulating PSD95, they had an effect increasing the cytoskeleton, but among all of them again, DHA surprisingly increased MAP2 levels significantly compared to the rest. So looking at these results, they were both surprising and exciting because we were looking at this compound expecting it to increase, but not to that extent that it actually outplayed all the other players. And it is even more intriguing knowing that DHA is a nutrient that is present in our diet. So with that in mind, DHA, as I already mentioned, it's a long-chain omega-3 fatty acid that is commonly found in fatty fish, such as salmon. It's also the major component of prenatal formulations, as well as the brain health supplements. It's part of 10 to 20% of the total lipids that are found in the brain, as well as over the 90% of the omega-3 fatty acids that are included in the gray matter. Among all of these, omega-3 of DHA also plays a key factor on the synaptic effects. So it is not just the availability of DHA that is compelling, it's also its biological relevance that caught our attention. So DHA is not new in the neuroscience research. DHA has been proven to increase synaptic scaffolding proteins, also upregulate PSD95 levels in both the hippocampus and cortex, as well as enhancing learning and memory behavior. So having all of that it also matches with DHA's effect on increasing membrane fluidity, as well as stabilizing all the receptors that we already discussed, the mTOR and the AMPR functions. And it also helps with PSD95 palmutylation, which is a key step where PSD95 is anchored into the postsynaptic region, just as Dr. Rami showed. But in real-life settings, DHA is not found alone. It's often consumed with EPA, another omega-3 fatty acid that is essential. Mostly of its functions is on the mitochondrial and also lowering the excitotoxicity in neurons, working as an anti-inflammatory marker. It actually has been proven that 1.8 grams of EPA in combination with DHA can help to significantly drop stroke recurrence in patients with high cholesterol. So having that in mind, we come with a question like, do different DHA, EPA combinations or ratios affect DHA's ability to enhance PSD95 expression? And if so, through what mechanism? So to answer this question, we designed another set of experiments. We use, again, our differentiated SHINE model, but this time we use DHA alone, as we used before for the screening, but we added what we normally found both either in the nature or in supplements, different combinations of DHA, having one that is predominant for DHA over EPA, one that is more balanced, and the other one where we have a more EPA presence than DHA. For one of the assays that we're looking at, we look at the cell viability, just looking at how toxic these supplements may be for the cells, as well as looking at immunofluorescence for our major synaptic markers, and confirming this with Western blot, which measures the quantification of protein expression. So what we found here is that, once again, DHA was able to increase PSD95 as it's done before by itself. But what was interesting is that when we added EPA, we got mixed results. So when we have a predominant DHA combination and we have an EPA predominant combination, PSD95 levels increase significantly compared to control.
Sorry, real quickly.
Yes.
Is this in wild type or haploinsufficiency?
So this is a wild type type of cell. So we're just looking at the beginning just to see if we can have an increase on the protein itself with a neuron-like cell. So just to flex, as you can see, our Westerns, they look very clean. Just saying. So as Dr. Rami did, we also were interested in, since PSD95 helps with the clustering, recruitment, and function of these glutamatergic receptors, we asked, okay, what does DHA does with them? So this is what we got. Looking at DHA by itself, it was capable of increasing both receptors, AMPER and EMDAR. But once more, when we add EPA to the equation, we got this modulation. AMPER receptors were significantly upregulated. They increased more than control at a balance of predominant DHA and a more balanced EPA-DHA ratio. But when looking at the EMDAR, the only combination that had a significant increase was a more balanced 1-to-1 DHA-EPA. So this can suggest that EPA can competitively modulate DHA effects in both the PSD95 and also on our glutamatergic receptors.
Sorry for,
Yeah, no
sorry
worries.
for interacting here, but are you saying that it's increasing synapse, the AMPAR, and is that a synapse response?
Can
you repeat the question for the people in Zoom now?
Yeah.
Of course. So the question is that if by looking at the glutamate receptors, we are looking at an increase on synapse, correct?
Development, what, by definition, what do you mean by AMPAR and NMDAR?
Okay, that's a fantastic question. So these receptors, what they do is like they are when you have the communication between the presynaptic and the postsynaptic cells, the neurons that are talking to each other, AMPER and EMDARs, what they do is they receive the message from one of the one given the message. They receive it to the neuron.
So you're not saying that there's more greater synapse development. What you're saying is there's greater synapse communication.
Yeah, we're increasing here the communication, the signaling between the.
Thank you.
Not necessarily the synapse. No, you're welcome, and thank you for the question. So here we have a summary table of our findings. So start with DHA alone. We saw that there was significant increase on PSD95 levels. Also increased both of the receptors, AMPER and EMDAR, making it a perfect candidate for increasing the protein scaffoldings as well as the glutamatergic receptors. When EPA was added, we got a different picture. So, So we increase, we can see that the balance, the combination of 2-to-1 increased significantly PSD95 and AMPER, but the EMDAR remain unchanged. And when looking at the more balance, we saw no change in PSD95, but increase on both receptors. So what this tells us is that DHA EPA or EPA added can have it does have a synaptic profile, but when we compare it to DHA alone, it's not as potent as just having DHA. But interestingly, when we have a predominant EPA, there's an increase on PSD95, but the receptors remain the same. So what does this mean? So it means that there's a disconnection between the structural upregulation with PSD95 with the receptors. So as a takeaway from this first part of the talk, some of the summary, so we can see that the DHA-EPA balance is highly sensitive, meaning that even the slightest changes in this ratio can alter the impact of DHAs on PSD95 and the glutamatergic receptors. And the EPA may modulate DHA synaptogenic potential through competitive or downstream mechanisms. So now that we know that DHA is a strong candidate, we ask a little bit more in-depth question, because we know that it changes at the membrane level, but what happens intracellularly? So that was exactly the question we asked. So what pathways can be involved that allows DHA to increase PSD95. So from the literature, we know that DHA doesn't work alone. It integrates to the neuronal membrane. It helps to stabilizing the receptors, clustering and anchoring. But we suspected that it may trigger some intercellular pathways that can increase PSD95. So when looking at it, we found that DHA can help with the neuronal survival and morphology through different mechanisms. So out of this picture and with a lot of different markers, the one that I want everyone to pay their attention on is this one here, which is the PI3K AKT pathway. It's a mouthful, I know, but bear with me. I'm going to be having trouble saying that over and over, so let's try to do our best. So for this one, what we can see is that this pathway, what it does, it increases, it phosphorylates AKT, which helps with synaptic plasticity, which is the brain's ability to adapt, learn, and create new connections. And this is important because it helps increasing PSD95 levels in neurons. So for that, we decided another layer on our experiments when we were looking at investigating the PI3K AKT dependent regulation of PSD95 by DHA and EPA combinations. So for that, we used a pre-treatment half an hour before with an inhibitor for the pathway. And after those 30 minutes, they were co-treated with DHA and EPA different combinations. We used the same measurements as before because we wanted to compare it with what we saw without the inhibitor as well. So we used the immunofluorescence Western blots, but for both the PSD95, MAP, and the receptors, but we also included the phosphorylated AKT, which is a marker that tells us if the pathway has been activated or not. So what we observe here is that the inhibitor by itself was capable of reducing PSD95. The inhibitor of the pathway reduced PSD95 levels significantly compared to the control, meaning that this pathway may be involved in the production of PSD95. But when we add DHA alongside the inhibitor, the PSD95 expression wasn't recovered. But interestingly, when we included EPA, we saw a moderate but visible increase on PSD95 levels, particularly at the more EPA versus DHA. This can suggest that it might be another pathway that can be upregulated with the inclusion of EPA. So on your left, we can see again the neuronal health marker, MAP2. Once again, we saw a reduction of this marker, that it wasn't able to be recovered when DHA was added. But we did see an increase on the 2-to-1 and 1-to-1 balance, DHA-EPA. But at the center, the graph that is here, for the AMPA receptors, it shows a more robust and interesting data. We do see that our inhibitor was capable of bringing this receptor down, but when we added DHA and EPA combinations, levels of AMPA were able to go back to the baseline or even above the control, meaning that this receptor may be more susceptible to a lipid-mediated recovery from the inhibition of this pathway. Lastly, the last graph on the right, however, we do see a decrease of the MDR receptors again when the inhibitor was present, but we do see, although we do see an increase on its levels with DHA and the DHA-EPA combinations, it didn't go back to the baseline, meaning that this receptor might not be as sensitive as the AMPER one. So here we're looking at the AKT. As I mentioned, it's a key marker to know if the pathway that we're interested in is actually activated or not. So when looking at total AKT, it was a small reduction, but not significantly. Looking at the total expression of this marker, when the inhibitor was added, meaning that the inhibition is at the phosphorylated level, as we're going to see in a moment. I'm going to explain in a moment, you can actually see it, but not at the total protein. So as I just said, when the inhibitor was present, the phosphorylated AKT significantly decreased compared to control, meaning that this inhibitor was capable of inhibited the pathway. When DHA was provided, there was a slight increase on this phosphorylation, meaning that it might be a partial recovery. However, it wasn't significant to bring back these, the AKT to a baseline. But once again, when EPA was added, we can see an increase on this on levels of this marker, meaning that AKT regulation may be mediated by another mechanisms through APA. So this is the mechanism that we're proposing that can be or may be happening in the cells, is that once we treat the cells with DHA, there is an activation of PI3K AKT pathway by activation of the phosphorylated AKT, increasing PSD95 level proteins as well as the receptors. However, when the inhibitor was present, there's an inactivation of the pathway as expected, but also a decrease on PSD95 and MDR levels. So, what's next? As everyone has been doing here, we've been trying to do the same, working with moving from our shy-Y model to a more relevant model using iPSCs. These are current pictures that we have from our differentiated neurons from a control cell line of iPSCs that we have, where we already tested them with DHA and with the inhibitor, and it has been the trend here. I just got these results this week, so I haven't been able to test them. But just visually speaking, we can see that PSD95, which is the green color, is less intense, and the MAP, which is the yellow color, is also reduced when the inhibitor was present. But what is more, I think, in my opinion, more interesting now, also another step that we're having, is that we are moving to a pilot clinical trial. And I know that for the genetic therapy, they need to wait for FDA and stuff, but fortunately for us with the DHA, since it has been proven its safety and almost non-side effects, we're going to be able to move ahead and try it. So for that, we're going to be recruiting patients for 3 months. It's going to be a phase 2 open label feasibility clinical trial, and that's because the number of participants that we're expecting is very low. So we're going to be just looking at the market without being able to provide clinical trials, statistical differences. We're going to be looking at null DLG4 mutations, meaning that it's just a loss of function. And we're going to be using a pharmaceutical grade DHA, and we're going to go with as high as 5 grams a day, because DHA has been proven that the longer the exposure and the higher the concentration, the higher or the greater the results we can observe in the brain. The intervention is going to be done for 12 months, a whole year. And these are the analyses that we're going to be performing. So we're going to be looking at baseline, 6 months, and 12 months. We're going to be measuring a cognitive and behavioral analysis. We're going to be doing an adaptive behavior, cognitive abilities, language development, and comprehension skills. We're also going to be performing MRI scans, looking at brain structure. We're going to have a general health panel looking at the full panel for a blood sample, but also we're going to be looking at the blood cell membranes, looking at omega-3 index, where we can see the total DHA and EPA levels. Finally, we're going to be also doing some multiomics. We're going to be using proteomics to look at PSD95 levels and lipidomics to see if DHA is going through the brain and if this is happening, how is it happening? So if it's DHA is incorporating to the cell. So just to finish, I think I'm on time. So we're going to have a few takeaways. Our in vitro findings suggest that enhancing PSD95 may support synaptic health, as we saw by increasing PSD95 and the MAP2 markers. We demonstrated that from our screen of 18 different compounds, DHA was capable of increasing PSD95, and that's due to its safety and known effects. We're going to be moving to a clinical trial. So it is thanks to your help and your support that we're able to move one synapse at a time. So I would like to thank Dr. MacKenzie for the confidence and the vote of trust of leaving me with this project. Dr. Zimmerberg for the amazing work with looking at the microscope, and our lab members, but especially to the SHINE Center Foundation for letting us work with these amazing people and family that I'm able to meet for the first time, and hopefully not last, the CHEO and the Precision Medicine Institute for their collaboration with the list.
Wonderful. Great stuff. Okay. I'm sure there's okay, one quick one. Okay, just okay. And poor kid has been reading.
Yes, and I can get Dr. Medina later, but did I understand correctly from all your slides that you were saying DHA with EPA is a bad thing?
It is not a bad thing. It can be competitive in reducing some of the markers with EPA, but when we're looking at the expression of PSD95, at least for this particular syndrome, we're going to be looking for a higher DHA. Because, again, as I mentioned, EPA has an anti-inflammatory response. So we do want to have EPA in our diet. So, and as we can see, we can increase PSD95 levels and their glutamate receptor levels. So it's not a bad thing. It's just that DHA can increase it higher or
Then with the combination
than the combination itself. But that doesn't mean that EPA is going to have a bad effect.
Okay. Just one question.
Just quick one. So this is SH cells. These are neuroblastoma progenitor kind of cells. Did you differentiate them when you did?
Yes.
Or it was done on the static?
No, that's a great question. And we do differentiate them. We use, there is a protocol that uses retinoic acid that helps them to differentiate for 7 days. But those are still immature neuron-like cells. So we added BDNF and the media that it's been sold by a company that allow us to obtain mature neurons as well. So, and we were able to, and that's something I didn't show, that when looking at the markers, there are some parts of the morphology of the cells that you can actually look, but also when looking at PSD95 and MAP, the levels of PSD95 and MAP were significantly higher compared to non-differentiated shyways.
The reason I ask is you don't see this puncta, which is really
Correct.
this hallmark of PSD95.
Correct.
But
Just real quickly, what would be the functional reason you'd see more PSD95 as a result of the introduction of DHA? I thought I saw some slides where you say we see more PSD95 protein. I mean, we have a gene that produces PSD95. Why, with the introduction of a supplement, DHA, would you see increase? What's the functional mechanism that would increase
Pathway activation.
That
but
would be
it's the
my-
protein we're talking about, not that.
But this pathway is going to help, at least in our, that's what we infer, we're thinking that the part of the gene that is working is going to be able to keep, it's going to help to bring more PSD95. So we're going to explode pretty much the
So it would
working gene.
upregulate existing genetic function?
Yeah.
Yeah, the normal gene allele is the drug target.
Right.
And that drug is making that gene.
So the normal gene allele, the argument here is through a supplement, we can increase the production of
the
good protein.
PSD95.
Got it.
Yeah.
Thank you. That wasn't.
But both go up, actually, yes.
Yeah, I was just curious, the same question. Is it like stabilizing the mRNA from the wild type allele, and that's why there is more production, like the mRNA is not being turned over?
That's a really good question, and that's something that
I
we
feel
are.
like throughout the talk I've been thinking, how is it even increasing? And
Yeah.
AKT is a kinase. It will phosphorylate. How does AKT bind DLG4 somewhere upstream for the expression? Anything in that pathway?
That's something that
We'll get back to you.
we still need to look into more in
either it
depth
is-
on the upstream and downstream for the pathway.